Hello. I don't know if you can all hear me. I'm getting a lot of reverb now. Oh, good. It went away. Greetings. Well, thanks for joining us. We are very happy to celebrate with you today. Obviously, we have all kinds of silly little things to remind us of the plus sides of this crazy time. And there are some pluses. None of you had to get on airplanes to join us today. And a lot of money was not spent unnecessarily. And you will see we're going to get a lot done in a short amount of time. And we're so pleased at the team that has come together to reinvent our annual event, which for 15 years has been a signature in-person event. And obviously, we're not doing it that way this year. So we've renamed it too. This came from the wife of one of our patients to call it a Merkel CELLebration. And I think that's going to stick well after COVID. But one of the key messages we have for you all today is COVID is not stopping us. It shouldn't stop all of you from doing a bunch of wonderful things, and it is definitely not stopping our team from making a difference in the lives of Merkel cell carcinoma patients. And today we wanted to bring you all together to help us supercharge that effort even more, because the way that we make big progress is to get ideas and collaboration and friends and resources and all kinds of things together. So we'll be working hard to have you all have an enjoyable time and also learn a lot and help us make a difference for patients. So today in terms of Zoom, I'll apologize. Boy, that recording was not like what it had been before. I think if you listen to it on our website now, and we'll have the links, the musicians will be playing at the same time as the music. Apparently having a few hundred people on audio throws off the technology a little bit. So we do have a few things to learn in terms of how to get Zoom to fully behave. So please mute yourself in an ongoing way until the breakout sessions, when people will be encouraged to talk and give their ideas. You can, I think most of you are, including your name if you desire, so we kind of know who's here. This meeting is obviously being recorded, and for questions, feel free. People have figured out the chat for the most part. Go ahead, put specific questions in there, or save them for the small group discussions at the end, which we will hope very much will be very interactive. So let's see here. This is our team that has put together this Merkel CELLebration. This is how we have worked. You guys can see that I'm at home. This is where I have spent most of my days for these past months. And I hope I can convince you that we can all be very effective when at home in a lot of cases. And my mom's always asking me, how are you getting work done from home? So hopefully this is she's on. Hopefully this will give her a little sense of that. And this is how we get work done here in bigger groups like this and smaller groups, and we can get a lot done. And we all go in on days we're happily not stopped from going into the lab to do whatever we need to do. Here, Nikhil and Alex are assembling the 240 care package info boxes that went out to many of you who made the request for those fun little things. And we got 340 people registered, and this whole team here has made rapid work of this reinvention of the annual event. So we hope you like it, and I want to thank all these people for their efforts in reinventing that. So our agenda for today is to go through a few scientific and clinical updates at the beginning, and then we will have two patient stories. Sue and Scott have recorded short summaries of their experience and some lessons and comments they have for Merkel patients. Then we will have a short virtual lab tour to show you a behind-the-scenes look at how we're getting things done. Then as we get ready for the small group discussions around 4:40 our time, we'll take a 10-minute break. And Krista and other people will put people into each of 6 rooms, and you'll be kind of randomly assigned into a room where there will be patients and our pharmaceutical partners and clinicians and scientists and everybody to kind of come together and think about how we can make the best possible difference, the biggest possible difference in the lives of Merkel patients going forward. So here's my one slide about Merkel cell carcinoma. All this information is available on our website, merkelcell.org, but around 3,000 patients per year are getting this cancer in the United States. This is an incidence that is rapidly increasing, and around 40% of those people will at some point have their cancer come back someplace, maybe right next to the primary, maybe in the lymph nodes, maybe far away. We can always make a dent and deal with that. We can always treat it, but this is more aggressive than melanoma. And what we'll talk about today is a whole bunch of progress in how we're detecting and dealing with those recurrences, and in some of the situations where we still can't do what we ideally would like to do. The things that are associated with getting this cancer are older age. More than almost any other cancer, this is associated with going up after the ages of 50, 60, 70. And sun exposure, fair skin, not having a lot of pigment in the skin, and immune suppression. In about 5% of Merkel patients, they have a real problem with their immune system. So 1 in 5, 20% of Merkels are caused by sunlight only, and 80% are caused by the Merkel cell polyoma virus, a crazy virus that's on our normal skin from the time we're little kids. But only in 1 in 3,000 people over their lifetime will lead to this cancer. We now have a pretty good understanding of the unlikely events that cause that to happen. And now we have a very good sense of how to treat this. With surgery and radiation, we can eliminate all the cancer in almost everybody that we can detect. And what we're going to talk about a lot today is after the cancer that we can detect has been eliminated, how do we stop it from coming back? And how do we detect it if it's coming back early? How do we find it early? So those are a couple of the major points. And then the final major point is, how do we deal with it if and when it comes back, and especially if it's then resistant to our best new immune therapies. So those are things we're going to tell you some exciting highlights about today. So I'm not going to detail a lot. The 2 FDA approvals in 2018 and 2019, the first ever leading to therapies for Merkel cell carcinoma recognized by the FDA, but most of the patients that we treat like that, who develop advanced disease, do really well with immune therapy. But eventually, it may stop working, and in the end, less than 50% have persistent benefit over years if they have advanced disease. So we're working hard to find better ways to get patients that long-term benefit over time. So there's the one warm-up slide about the concepts, and I'm going to tell you how we're organizing everything today is to talk about the patient experience in these 5 steps here. A patient obviously initially has a bump or something. Somehow there's a diagnosis that's made, and then they undergo initial treatment, often with surgery or radiation. And then we start watching them after we get rid of all the cancer we can find. And we'll usually watch folks for up to or around 5 years. And most of them, happily, will not have a recurrence. But for those who do have a recurrence, we want to be able to find those recurrences earlier. And you'll hear several stories about how we're able to find those earlier now and recent progress in doing that, as well as if it comes back in places we can't just treat with surgery or radiation like the first time here, how do we get rid of it? How do we control it? And we have a lot of progress in using the immune system for doing that. And one of the biggest challenges we face is how to treat it if the immune drugs that are FDA approved no longer work. So we're really excited to give you updates on all of those fronts. So we've kind of broken this down now by, and we are having a background problem. Perhaps that person, I don't know if you're hearing it, but that person, we could maybe pop them out and then they could rejoin or something. Okay, so now can you hear me now? I unmuted myself. Okay, the joys of Zoom. All right, we keep learning. So Aline Fonseca is going to talk to you about progress in this first phase here, the initial treatment phase, where we now know that we need to get a scan on essentially every Merkel patient at the time of diagnosis, because if we don't, we miss actually disease that you can't feel or see that has jumped somewhere else in the body in 1 in 8 patients. And that means we mistreat completely 1 in 8 patients. So happily, we've studied our population, all of you in many cases, and we've learned this, and this is rapidly turning into international guidelines for better treating this cancer. Then Dr. Fonseca will also talk about the concept of surgical margins, which means how big does the surgeon cut around the obvious tumor? Do they go just barely around it, or 1 centimeter or 2 centimeters around it? And that turns out to be a very complicated thing, but we found a simple solution, and she's going to talk to you about that, and that really helps patients have fewer side effects and control their cancer better. Then Dr. Pete Goff is going to talk about single fraction, that's kind of like single dose radiation, which we're using increasingly. Instead of giving people 6 weeks of radiation, we're often able to give them only 1 dose. It's a bigger dose, but that seems to be working in the case where the surgery appears to have taken out the cancer, and we're just trying to have an insurance policy that it won't come back. So he's going to talk to you about that exciting update, which really can affect many patients. Lauren Zawacki is going to talk to you about the antibody blood test that helps us to detect the cancer when it's coming back earlier, and how that's making big progress. And then Kelsey Cahill is going to talk about another thing that affects patients who are in the surveillance period, and that is a recurrence risk calculator. Right now, we mostly only use a person's stage at diagnosis to talk about how they get treated and how often they get seen and how long they're seen for. And Kelsey's going to talk to you about how much this has improved now using a new calculator that's available now on our website. And Dr. Cherney is going to talk about the very exciting concept of a therapeutic vaccine. We are now working with several companies that are keen to try to explore whether virus-positive Merkels, and that's 80% of them, could be prevented in coming back by giving a vaccine that increases the immune response against the virus and thus against the cancer. So then we'll come back to Dr. Fonseca, who will talk about 3 clinical trials that affect early patients where we're trying to prevent the cancer from coming back, and then trials that are in patients who have already had their cancer come back even after immune therapy. And we're happy to report that there is real progress in that very tricky realm as well, where the cancers come back and they stop responding to those drugs that you all know as Bavencio or Keytruda, those kinds of drugs. And then what do we do? That's been the biggest problem, and we are making progress in that realm, thanks in part to support from the NIH, which is helping us lead a trial that Aline is going to tell you some happy updates from. So I'm going to turn this over now to Kelsey, who's going to talk to you about the recurrence risk calculator.
Thank you, Dr. Niam. And hello, everyone. My name is Kelsey. I'm a research coordinator with the lab, and I'm excited to tell you all a little bit about our recurrence risk calculator, which you can find on our website at merkelcell.org/recur. And so we're talking about the chances that someone's cancer comes back. So we're right here on the surveillance part of our timeline. And I'm sure many of you have seen a graph that looks like this before. And what it's showing us is the percentage of patients whose cancer did not come back following a certain period of time after their initial treatment. And this black line corresponds to all stage 3A patients. And stage 3A means that these patients have had their cancer spread to their lymph nodes. And the recurrence risk estimations that you usually hear, such as this one, are only considering stage. So when we consider nothing else about a stage 3A patient, we would estimate that the chances of their cancer coming back is 41%. However, we now know that there are other factors that can influence this risk to be either higher or lower. And these factors include sex, because we have found that women are 40% less likely than men to have their cancer come back. And next we have age at diagnosis, and this doesn't have quite as big of an impact as sex, but younger patients do tend to have a lower risk. And next is the site of the primary tumor. Some sites carry a higher or lower risk than others. And lastly, we have immune status, because the immune system plays an important role in controlling this cancer. So individuals who have a compromised immune system would have a higher risk. And so our calculator takes these factors into consideration and is based on data of over 600 patients. And those 600 patients actually include many of you who agreed to be a part of our research and who take the time to answer our emails every year to let us know how you're doing. So thank you. This tool wouldn't be possible without your help. And so when you use the recurrence risk calculator, a graph that looks like this will be generated. And interpreting these graphs can be a little tricky. So what you want to pay attention to is how far down the line goes before it starts to flatten out. Because remember, this axis is looking at the percentage of patients whose cancer did not come back. So the further down a line goes, the higher the chances are that their cancer will come back at some point. And so we're going to show you a couple examples of different patients to show you just how much this risk can vary when we consider these other factors. And first, I want to acknowledge a few of the people who were involved in developing this tool. And so in our first example, we are looking at a stage 3A patient who we'll call Mary. And a few of these factors are actually going to lower her risk. So she's a woman, and she's also a little on the younger side for a Merkel cell patient. So we expect her immune system to still be fairly strong. And she also has an unknown primary. And what that means is that her cancer was found in a lymph node, but there was no detectable tumor on her skin. And when it comes to cases like these, we think it's likely that the person did have a tumor somewhere on their body at some point, but their immune system detected the cancer and got rid of it before it was even noticed. And we believe individuals who have a robust immune system like this are less likely to recur. And so when we take these additional factors into consideration, we find that the chances of her cancer coming back are quite a bit lower than if we were to only consider her stage. And so Mary falls on this blue line, and we'd estimate there's a 23% chance that her cancer will come back. And so her doctor may decide they don't need to be quite as aggressive with her treatment and surveillance. So in our next example, we have another stage 3A patient who we'll call John. And some of these factors are going to increase his risk. So he's a man, he's a little older at 85, and his immune system is suppressed because he has a type of leukemia called CLL. And CLL is common in older people, and while it's not very dangerous on its own, having Merkel cell at the same time can be much more serious because CLL suppresses the immune system, and the immune system plays a very important role in controlling this cancer. And so when we take these additional factors into consideration, we find that the chances of his cancer coming back are much higher than if we were to only look at his stage. So John falls on this red line, which we see goes way further down, and we would estimate that the chances of his cancer coming back are 82%. And so his doctor may decide that they need to be a bit more aggressive with his treatment and follow up with him more closely and for a longer period of time. So in both examples, we saw that the risk of recurrence is either much higher or much lower than if we're only looking at a patient's stage. And so we hope that this tool will be helpful for patients and clinicians by giving them a recurrence risk prediction that's more individualized for the patient so they can make a more informed decision about treatment and surveillance. And now Lauren Zawacki, a research coordinator with the lab, is going to tell you all a little bit about the AMERK test.
Thank you, Kelsey. My name is Lauren Zawacki, and I'm a research scientist at the NEOM lab. And one of my main roles at the lab is coordinating the AMERK serology test for Dr. NEOM's patients. And the AMERK test was actually developed in the lab several years ago, And the individuals who played a large role in the development and implementation of this test are shown in the bottom right-hand corner of your screen. So the test was also developed in collaboration with the University of Washington's Department of Lab Medicine and became clinically available in 2014. So the AMERK test actually measures the amount of antibodies in a patient's blood. And as you can see on your screen, if a patient produces antibodies at the time of diagnosis, then there will be a large amount of antibodies present in the patient's blood upon the initial diagnosis of MCC. And after the initial treatment, which includes things like serology and radiation, the patient's antibodies will decrease, and the patient should continue using AMERK in their surveillance plan. However, if there is an MCC reoccurrence, these antibodies will increase again because there is presence of new disease. So to summarize, antibodies will rise when there is new disease and remain stable or decrease when their cancer is going away. So therefore, we suggest that this test is done after a patient is diagnosed and is used to monitor the disease after the initial treatment. And once the test is done, patients will fall into one of two categories: patients that either make antibodies and those who do not. And only about 50% of patients will produce antibodies at the time of diagnosis. So for patients that do not produce antibodies, we know that we cannot utilize this test and that these patients have a higher overall risk of their cancer coming back. And therefore, we must follow these patients closely with scans. However, if a patient does produce antibodies, we can utilize this test, and it's very beneficial in this case to know if their cancer is coming back. And typically, we use less scans. And therefore, this is why we believe that this test is useful for all MCC patients upon diagnosis, because it determines a surveillance plan for a patient. So using the AMERK test and having fewer scans has several advantages. And these advantages include that there is no contrast IV injected into the patient, the AMERK test is less expensive than scans, and the AMERK test can oftentimes detect MCC disease earlier than scans. Furthermore, since our lab developed this test about 7 years ago, it has been adopted nationally. In 2020 alone, it has been utilized in over 100 centers in the United States, which you can see on your map right here. Additionally, in 2019 alone, there was almost 2,000 tests that were performed in the United States. The test was even adopted into the National Cancer Guidelines in 2018, and these guidelines are what doctors reference in determining how to treat a patient. And you can see that there's an increase between 2018 and 2019 right here on your graph, and we believe this is because of the changes in those guidelines. We believe that the continual increase in the AMERK testing will allow a larger number of MCC patients to benefit from this test. And at this time, we would specifically like to thank the University of Washington's Department of Lab Medicine because they receive and process all of these blood samples. And without them and their team, this continual increase would not be possible. I would now like to introduce Dr. Goff, who is a physician scientist in our lab and who is particularly interested in the use of radiation therapy in treating MCC patients.
So thank you, Lauren. I'm a resident in radiation oncology, and today I'm going to talk to you about some of our recent efforts at University of Washington to optimize how we use radiation therapy to treat MCC. It's an important treatment at all stages of MCC, but it does cause both short and long-term side effects, and we need to judiciously use it in order to maximize its benefits for our patients. So one of the most important places that we use radiation is following a successful surgery in which all of the visible tumor has been removed to kill the few remaining cancer cells that are left behind in some patients in order to reduce the chance that the cancer will come back at the site of the surgery. So in this way, it plays an important role in curing the disease in its earliest stages. As physicians, we cannot predict whose cancer will come back at the initial surgical site. However, we know that in the majority of patients, tumors do not recur locally just with surgery alone. However, and unfortunately, this means that about 20 to 30% of patients are at risk for their tumors coming back at the site of surgery, even after a successful surgery. However, this number is reduced to less than 5% with radiation. So we end up recommending radiation to all MCC patients. Now, unfortunately, this means that many patients who get radiation don't have any benefit from it, and it does have significant potential to cause harm. So the treatment that we typically recommend is 25 daily treatments, which are given spread out over 5 weeks, as you can see here, for standard radiation after surgery. And each one of these doses or radiation treatments is given at a dose of 2 gray, which is a unit of radiation, every day. Now, these can cause side effects, especially in the sensitive head and neck region where many Merkel cells occur. And this is things like skin burns, dry mouth, and dental problems. Some of these go away soon after the treatment is over, but some of these can persist and cause a decrease in patient quality of life. When a patient with advanced Merkel cell carcinoma, often without any good treatment options, has either one or more tumors that is either painful or in a dangerous location, we often just give them 1 dose of radiation. This is a larger dose, which is just given on 1 day, and something that we term single fraction radiation therapy. This tends to be very well tolerated and very rarely has long-term side effects. Dr. Iyer studied the response of MCC treated with a single fraction of radiation, so just 1 8-gray dose, in the setting that MCC had spread throughout the body. And what she reported about 5 years ago is that MCC is very sensitive to radiation and compared to other tumors, with 94% of tumors responding to just this single dose of radiation. So this prompted us to ask whether this single 8-gray dose, which we know is very well tolerated in comparison to the standard treatment course of 25 fractions, can be used after surgery in order to reduce the risk that the tumor comes back following a successful surgery. And so that question was looked at recently, and the initial results were published by several of our investigators here at University of Washington. So Matt Cook and Dr. Schaub and Dr. Parvathanini, who many of you know as Dr. P, have looked at this and published our first small series, which is 12 patients. And what they saw initially, and this is something that we've understood well, is that this single fraction radiation at 8 gray is very well tolerated. There are very few side effects as we expect compared to the conventional 25 treatment course. But very importantly, we didn't see any recurrences in patients that have been followed in this initial group of 12 patients that we've followed after single fraction radiation therapy. So this is very promising early efficacy. We've offered this to a number of patients at University of Washington. We have dozens more that we're following closely. We think this offers a superior patient experience with less side effects, much less time on treatment, and we feel fairly confident that this is working. So we are routinely giving this as an option to our patients here at University of Washington, but we are being diligent in continuing to study this in a longer group of patients for a longer period of time to make sure that it is equally effective to the standard radiation. We're going to start working on promoting this to the wider MCC community so that more patients can benefit from this treatment. And with that, I'd like to introduce Dr. Fonseca, who is a clinical research fellow. She's working on a number of projects. She is seeing patients with Dr. Neum in his clinic at the SCCA, and she's going to discuss several updates in both the clinical care of MCC as well as some ongoing clinical trials.
Thank you, Dr. Goff, for the introduction. Hi, everyone. I'm Dr. Fonseca, one of Dr. Neum's clinical research fellows. And so I am going to now tell you all about imaging, which is in the initial treatment portion of our timeline. So is a scan at diagnosis important for Merkel cell carcinoma patients, and why? So currently, unless a person's lymph nodes are obviously involved with Merkel cell carcinoma, in other words, you can actually see or feel the lymph nodes in common places, current guidelines recommend for most Merkel patients to not get a scan at the time of diagnosis. So we wondered if this was right. Is this best for patients? So we went to our data and we were able to see that the truth is among 492 patients, 1 in 8 Merkel cell carcinoma patients actually had their care completely changed if they got a scan before their surgery. So because of this reason, we now think that it is very important for all Merkel cell carcinoma patients to get scans before undergoing treatment. Otherwise, 1 in 8 patients will get the wrong kind of treatment, such as over-aggressively treating the primary site when in fact the cancer is already someplace else. So a patient who didn't get scans before undergoing treatment could get too much treatment to the primary site, which can have all kinds of side effects. And the distant site would be completely ignored because we didn't even know there was cancer there. This is very bad for the patient's real long-term outcome. And we were able to come to these conclusions, all thanks to many of you for participating in our research, allowing us to see how you all are doing. That's been super helpful. And you can hear throughout the whole day today, it's been changing the way this cancer is managed. So now I'm going to talk with you all about some more concepts that are also in the initial treatment segment of the timeline. So optimizing surgery and radiation. So these are pictures of a narrow surgical margin and a wide surgical margin. So surgical margins means how much normal skin the surgeon should cut beyond the tumor. So this picture is showing a 1 centimeter margin, which is narrow. And this picture is showing a 2 centimeter margin, which is wide. So the wide margin, if we add 2 centimeters, and then this 2 centimeter tumor, and another 2 centimeters, we get a 6 centimeter hole. Versus with the narrow margins, where we add 1 centimeter margin, this 2 centimeter tumor, and then another 1 centimeter margin, we get a 4 centimeter hole. And this becomes a 10 centimeter incision, which I will explain to you more shortly. So here you can see that a wide margin ends up becoming a 15 centimeter incision. This is because surgeons can't close a circle or golf ball size excision. They instead remove tissue in a football shape to bring the tissue together. So 10 centimeters in the narrow margin versus 15 centimeters in this wide margin actually turns out to be a much bigger difference than you would think. So the incision needs to be 2.5 times larger than the height. That's why this 2 centimeter wide margin turns into having a 15 centimeter incision, because 2.5 times 6 centimeters is 15 centimeters. That's big. 16 centimeters on certain parts of the body is either impossible to do, or when you can do it, you have a big defect. You may possibly even have to get a skin graft, and this takes months to heal. So if surgery with very large surgical margins, that can be very hard on the patient. And we give lots of radiation. If a patient also needs lots of radiation, that can be very hard on the patient as well. So we needed to figure out how to integrate surgery and radiation to both not have the cancer come back and not have patients have a bunch of side effects they don't need, such as problems with wound healing and treatment. So how big does surgery have to be? To answer this question, again, we went to our data, thanks to many of you for participating in our research, and found that in a new study of 148 patients led by doctors Tara Badkar and Fu, if radiation is planned, in other words, you know you are going to be doing surgery after radiation, then narrow margins are sufficient in preventing the disease from coming back and reducing bad side effects. So if you're going to do radiation, you can go to a margin that's basically 0. You can just cut out the tumor and then undergo radiation, and you do just as well as if you took a much larger area. So if you do a 2 centimeter wide margin, it becomes 15 centimeters. If you do a 1 centimeter margin, it becomes 10 centimeters. And if you do a 0 centimeter margin, you can only cut out the tumor. You can just close the incision, which is very easy, and then you can begin radiation shortly thereafter, and the effects are markedly reduced. So narrow surgical margins when radiation is planned is now being adopted and incorporated into the major international cancer guidelines used throughout the world on how to manage Merkel cell carcinoma. So now we will discuss a couple Merkel cell carcinoma adjuvant clinical trials. Adjuvant means that is where we give treatment where we think there's cancer left, but we don't know for sure, in order to prevent it from coming back. So these clinical trials occur after the initial treatment and continue in the surveillance segments of our timeline. So lots of patients have gotten rid of their cancer to the best of our ability through normal approaches like surgery and radiation. But unfortunately, there is still a 30 to 80% chance that their cancer is going to come back and it's hiding someplace. And we have no approved therapies to prevent patients' cancer from coming back. And the current standard of care is to watch and do nothing and try to catch it early if or when it does come back. So with these clinical trials, we are trying to do something. We are trying to determine if we can prevent the cancer from coming back. So there are 2 current clinical trials that use immune therapy to see if it can prevent patients from having their cancer come back. So we have the ATOM trial, which uses avelumab, in other words, Bavencio, or placebo, again, after initial treatment of high-risk Merkel cell carcinoma. And then we have the STAMP trial, which uses pembrolizumab, in other words, Keytruda, or observation after surgery. Both of these trials are being explored for whether they can prevent the cancer from coming back. And depending on these trials, it will really help us stop recurrences from happening. And now I'm going to talk about the hardest problem that our whole field faces, and that is, what do we do for patients when they don't respond to one of the approved immune drugs like Keytruda or Bavencio? We believe that part of the reason why these patients stop reacting to or benefiting from these immune stimulating drugs is they don't have enough or the right kind or functional enough T cells anymore. This is the biggest problem we face in our field. And now I am going to tell you all about Merkel cell carcinoma T cell trial, which occurs at the recurrence treatment part of our timeline. And here is the wonderful team of scientists who have developed this. So patients who can be a part of this trial are those who have virus-positive Merkel cell carcinoma and are no longer responding to approved immunotherapies. As you all know, as Dr. Neum has mentioned before, in 2018 and 2019, there were very exciting FDA approvals of immune therapy drugs. But there are far more people who no longer are responding than we wish. And if you don't respond to one of these drugs and you're virus positive, you might be eligible for this trial. So this clinical trial is called the ATTACK trial, and patients in this trial would continue their current approved immune therapy, which isn't completely getting rid of all their Merkel cell carcinoma, but we think might still provide some support to the immune system. And thankfully, with modern biology, we are now able to reprogram a person's own T cells to recognize and kill just Merkel cell carcinoma cells. And we just saw one of these patients that was in this clinical trial back in clinic last week, and I'm delighted to share with you all that we had some wonderful results from this patient who had stopped responding to Keytruda. So this is definitely encouraging, positive news, and this trial is currently continuing to enroll patients. Next, Dr. Kearney, who is a research fellow in the NEOM lab, will tell you all about a new therapeutic vaccine that we are creating.
Thank you. I wasn't able to unmute myself. Sorry about this. Thank you, Dr. Fonseca, and hello, everyone. My name is Shira Charney, and I am a research fellow in the NIAM lab, and I'm going to talk with you about the MCC vaccine. So just as Dr. Fonseca mentioned, after initial treatments, MCC patients don't have any other approved treatment. As Dr. Fonseca described, adjuvant therapies are given after the initial treatment when we can't detect the cancer, but it is possibly still microscopically existing in the body. The current clinical trials that are adjuvant therapies give immunotherapy to high-risk patients, which is an expensive drug and have a risk of side effects. But we are trying to figure out other methods that can prevent from the cancer to return and be safe for low-risk patients as well. We are working on a vaccine that will be given after the initial treatment and in the beginning of the surveillance period. So when we talk about vaccine, what most of you are familiar with is a preventive vaccine that is given to block infection of known viruses, such as the flu vaccines and those that currently being developed for COVID. But an MCC vaccine is a therapeutic vaccine that would be given to MCC patients after initial treatment. And we expect this vaccine to be very safe. So what we are looking here is on the amount of T cells that recognize MCC cells in the blood and the time from surgery. So what we know about the virus-positive Merkel patient is that when you get the surgery, there is a large amount of T cells that we can detect in the blood, and that these T cells can kill virus-infected cells. But as soon as the surgery or the radiation happens and the cancer goes away from the body, over the course of 4 to 6 months, as we see here on this time after surgery, the detectable amount of T cells goes down. And we think this is part of why the cancer is coming back. So the main goal of this vaccine is to maintain this robust anti-MCC immunity and to keep these T cells that recognize MCC cells patrolling the body and eliminating the microscopic disease that is present in probably about 50% of the patient and that we are currently don't have any way to address it. So we are happy to update that we are currently working with several different companies and different technologies to find a therapeutic vaccine for MCC patients. Like with COVID, we cannot predict based on my studies what will work best in humans. Therefore, we plan that all these technologies will reach human trials in order to find what works best for MCC patients. As of today, there is one platform that has been tested successfully in mice, and we will be moving forward to a clinical trial, hopefully in the late 2021. And with that, I would like to finish and pass the presentation back to Dr. Niam. Thank you.
Great. Can you guys hear me? Good, good. Thank you. So thanks everybody for giving those updates. Those are what we had selected today as things that I hope convince you that a lot of progress is happening. Those are all new updates this year and really reflect changes that are happening around the world because of the studies that have been getting carried out here. So we're very happy about that. And you can imagine all of that work took a lot of effort. And so I want to have a few thanks here, not the least of which is to funding sources. Over $20 million over the past decade or more has come into Merkel cell carcinoma research from the National Institutes of Health. So these are literally your tax dollars at work. In case you're looking for some good news about how your tax dollars are spent, we think this is very, very impactful work. We want to thank our many industry partners. There's no way we would be developing drugs, including that T-cell therapy drug, to bring to people if we didn't have industry partners as well as the NIH to get us going. We have many research collaborators around the country and really around the world, and over 50 ongoing projects, each of which is headed towards a publication at some point. So that's a lot to juggle. This is all made more possible by donations. And since 2006, there have been over 4,000 individual donations. The average is around $75. But in aggregate, that's a very impactful, almost $6 million. That is money that we don't have to spend on a precise schedule, that we don't have to spend on a proposal that we wrote 2 or 3 years earlier, and we can really carry out flexibly. So extremely impactful to have those gifts. We have many clinicians and clinical staff on our extended team. We now have a Thursday noontime conference every week where we have up to 20 doctors and scientists or even more coming together to talk about our trickiest cases and colleagues presenting to us from around the country. So it's a really excellent team helping to bring great care to patients. And then biggest thanks to all of you, the patients whose participation in research we've told you in several cases just by telling us how you're doing. That's enough to, in combination with what we know about the time you were diagnosed and how big it was and where it was and what your age was and all those things, is very powerful information. But also the blood and tumor samples are also incredibly powerful in setting up and allowing us to do all of these immune studies that have really brought about major changes in this cancer. So we have a couple minutes now to answer questions. I think we've got several that have come in on chat. I have a couple scribbled down that I think would be of interest to a lot of people. So I think we have about 10 minutes that we can do that. So you can keep the questions coming in from, on specific topics. And then we will later, if we don't get to them all, right now, we will send out a summary of questions and answers by email, so you'll have those. And in 10 minutes, then we'll take we'll switch modes, and we'll have 2 patient stories followed by our virtual lab tour. But I think we can take a couple questions now, and I have one. I know one that came in from several people is, what of those trials is possible in virus negative Merkel cell carcinoma as compared to virus positive? So both the T-cell therapy trial and the current trial that we're planning for a vaccine will start only in virus positive Merkel's. And that's because we have a simpler, more tractable problem there. We kind of know what the enemy is, we know what the target is. However, we and others throughout all of cancer research are figuring out ways in which all of those things will be possible in the future in virus negatives as well. It's just that it's more complicated because we have to go in and find customized solutions for each person's cancer, whereas we can do it in a more shared solution approach with the virus. So for right now, that is going to be done for virus positive Merkel's. So let's see, other questions? I don't know, Krista or anybody, if you want to just read out a question we can address.
Yeah, this is Krista. Hello, everyone. I received some private messages and some questions about immunotherapy, which we didn't talk about too much today, but some of the questions were, when is immunotherapy appropriate for patients, and what does the typical course look like? So how long does the patient stay on immunotherapy if they don't have a recurrence? And also, if they have a recurrence, do they stay on that immunotherapy?
Okay, well, who gets it is a good start. And basically it's for people where we can't just do surgery and radiation. And that gets a little tricky because in theory, some surgeons can do some really big surgeries. But so that's a customized decision. But actually more and more patients now are eligible for immune therapy, because sometimes if you just present with some big lymph nodes and a tumor in your arm and something like that, sometimes it makes sense to try immune therapy first. That's a very tricky discussion, but that's called neoadjuvant therapy, and that is increasingly studied and done. And so that's being done at least in some cases. And then Krista, was one of the other questions, when do you stop immunotherapy?
Yeah, so if a patient hasn't had a recurrence, how long do they stay on the drug?
So we don't have a perfect answer for that. We're working on that really big question very hard. If somebody has a side effect from their immune therapy, like their immune system starts destroying their own body, obviously we stop the drug right then or much sooner. Even if it's a modest side effect, we're more likely to stop it. But if you don't have any side effects and the cancer is well controlled, we never stop those drugs in less than 1 year, unless we're forced to. And we like to go more like 2 years. And then what we're shifting to now, because we've had quite a number of patients when we stop 1 or 2 years later, have their cancer come back. We don't really like that. Sometimes their cancer will respond to the same immunotherapy drug, and sometimes it will not. So what we're shifting to now, instead of sort of cold turkey, stop the drug, we will increase the interval and give it, instead of every 2 or 3 weeks, we will give it once every 3 months or something like that. And we'll do that for another year or two with very much less cost, less hassle, less side effects, and that's working very well. So I think those are some of the approaches that we're doing, but we're carefully studying this and we're collaborating internationally to get the best data about when and how to stop immune therapy.
Yeah, so some other questions that were pretty common were regarding the AMERK test. One participant asked, is the AMERK test now standard of care for Merkel cell carcinoma? That is yes. It is now included in the National Cancer Guidelines. And then let's see, another question was, this is kind of going back to the virus negative question, but what is the best way to screen virus negative patients for recurrences?
So we have an exciting update there that is one of the other 55 projects we didn't talk about today, but there is a new blood test that works particularly well for virus negative patients. And we just had our first patient really have benefit from this test. And it looks for DNA in the blood released by the tumor. And you first have to figure out and sequence all of the DNA in that person's tumor. And believe it or not, that's not a hard thing to do. And you find the unique mutations present in that person's tumor that are not present in the rest of the DNA in the body. And then you just look for those little snippets of DNA floating around in the blood. And it turns out that DNA from the tumor is only released from the tumor if there's actually tumor in the body. And DNA only lasts in the blood for a few hours, not months or years. So if we take blood out of a person and we look for special segments of DNA present only in the tumor, we're able to now test for the cancer coming back even in virus negative cases. So that's something we just in the last 2 weeks had our first positive from, and we fully anticipate using that more in more patients in the future.
Great. We probably have time for maybe 1 or 2 more questions. I want to switch gears to the single fraction radiation. So someone asked whether single fraction radiation is effective in high-risk patients. For example, patients that have immune suppression, such as an organ transplant.
And the question was, for high-risk patients, what, Krista?
Does single fraction radiation therapy work?
Yes, it does. It works exactly as well, interestingly, in the initial response in patients with and without a good immune system, but you're about 3 times more likely to have it come back, about 30% instead of 10%, if you don't have a good immune system. So this is something we're really excited about in general, is augmenting the immune system and helping to shrink the cancer in a way that turns on an immune response. And in your little brochure, we're talking about a DNA damage response inhibitor pathway where we think we're going to maybe be able to combine these things in a way that kills the cancer with a little bit of radiation, but also turns on the immune system, because we know that the immune system is very important in controlling the cancer in the longer term. And I saw a great question from Robert Marcus I can address easily there. It's kind of a fun one about the AMERK test more broadly. It's like, well, since it's standard, is Quest or one of the big labs gonna start getting involved? Well, honestly, we would love for them to be involved. Not only, they don't want to be involved. We're trying desperately. If anybody has any friends at Quest or something like that, we want them to partner with us, but they think it's too much of a niche market. They're a little busy with COVID right now as well. But we need some partners around the country to help this test go forward, and it's a complicated and a kind of expensive test to run. So we're just barely breaking even. Traditionally, we've lost money on it, and we're just now hopefully focused on making sure it stays available. But it is increasingly adopted, and I think eventually we'll have some good partners that'll make it more straightforward to run.
Yeah, so there's a lot of other questions people asked, but we are going to have a small group discussion where we can answer those questions. And we've also been taking notes of all the questions people have written, and we're going to respond to those and circulate those out if we don't get to them at the end of the meeting. So
I'm going to do one more quickie. I love it. What happens if you have an autoimmune disease and we're all too scared of using these antibodies that just turn on the immune system and can have really bad side effects? We are absolutely trying to focus our anti-Merkel effects so narrowly that we will offer safe treatments to such people. And in theory, those T cells that Aline talked about that we are just now seeing working in some patients, those can be focused in a narrow way. And in the future, we really hope and think that we're going to be able to have very targeted immune therapies that are not going to make your MS worse or your rheumatoid arthritis worse or something like that, and will deal with the cancer. So there's a lot of cool progress that's happening way beyond our team, let's put it that way. And we fully anticipate to harness it for our Merkel patients in the future. And that's really one of the things we want to do, is patients with immune problems, how do we help them with very targeted immunity? Okay, so I think we can now go on to